Assalamualaikum and good day, Doctor. My name is Khadija and my group member is Siti Afifah Ramli. So, we're going to present our topic on microplastic contamination in food. So, the contents of our presentation first is introduction, next is acute and chronic toxicity, next is route of exposure, symptom, metabolism, mechanism of action, and the last one is conclusion. So, as an introduction, so what microplastic? Microplastic is actually plastic that has mean size less than 5 micrometer, and it also can achieve size 1 to 1000 micrometer, and this known as a nanoplastic. So let's continue with the global issue and health concern. During 2018, about 359 million tons of different plastic material that has been used to produce plastic-based products, and these plastic-based products actually one-use goods. One we use, we can directly dispose, like plastic straw, plastic bags, and diapers, for example. So from all total of plastic-based products, only 21 to 26% of the plastic waste has properly managed. The rest falling under improper waste management. It has been discarded into the pit, into our environment, and leading to the plastic pollutions of the water, air, and soil. So because of this practice, this microplastic ubiquitous in our environment, and this will further enhance the exposure risk of microplastic on human. There are two sources of microplastic. The first one is primary microplastic and the second one is secondary microplastic. So primary microplastic that is manufactured in particles that are micro-sized, such as microbeads, plastic fibers, and plastic pellets, and all of these will be used to produce plastic-based products. For the secondary microplastic, plastic that result when large particle items break down, for example, tires break down, carrier bags break down, it will produce microplastic. Figure 1.1 shows the pathway on how plastic is converted into microplastic and nanoplastic. So from the figure, we can see that several types of plastic, PE, PP, PS, PVC, polyurethane, epoxy, alkyl, all of these can turn into microplastic through several types of reactions. As you can see from here, biodegradation, photodegradation, thermo-oxidative degradation, thermal degradation, hydrolysis, and abrasion. And all of these types of reaction can turn the plastic into microplastic. And this microplastic, uh, for several period of time, it also can turn into nanoplastic because it is exposed to the light, to the UV. And this reaction can even happen under moderate temperature, around 30 degrees Celsius. Now, I continue to the second part of presentation, which is acute and chronic toxicity. Oxidative stress is one of the acute toxicity of microplastic. So microplastic can bind to heavy metals, phthalates, BPA, and it can penetrate cell membrane and enter the cytoplasm. This will trigger the immune response that stimulate the production of free radical that leads to oxidative stress. For example, BPA can disrupt mitochondrial function, an organelle crucial for energy production in cells, leading to the generation of ROS. With excessive ROS production, the body defense system, for example, glutathiones, superoxide dismutase enzyme, becomes depleted, and this leads to an antioxidant deficiency, worsening cell damage, and organ due to oxidative stress. Chronic toxicity of microplastic, for example, systemic inflammatory. When ingest or inhale microplastic can cause inflammation in the body. GI tract. They damage the epithelial cell of intestines, disrupt the microbiota, and trigger oxidative stress in the GI tract. In lungs, if inhale, microplastic cause oxidative stress in the lungs, leading to chronic respiratory issue. Second chronic toxicity is neurodegeneration. That microplastic and the chemicals they contain can negatively impact the central and peripheral nervous systems, leading to disruption in brain and nerve function. Microplastic also can cause cancer. For example, BPA, known as an endocrine disruptor, it can interfere with hormone function and increase the risk of several types of cancer, particularly breast and prostate cancer. Phthalates used as plasticizers. They have been linked to an increased risk of liver cancer, kidney cancer, and breast cancer. Polycyclic aromatic hydrocarbons, carcinogenic substance that can be absorbed by microplastic and can cause skin, lung, and liver cancer when exposed over the long-term period. I will continue with the third part of presentation, which is route of exposure. Figure 3.0 show the exposure routes of microplastic contamination to human. First, ingestion. Second, inhalation. And it also can be exposed through dermal. And the major exposure routes of microplastic to human is ingestion. So, how can exposure through ingestion occur? As we know that most of the plastic waste has been discarded into the pit, into our environment, and this causes plastic pollution in water, air, and soil. So because of this practice, a lot of microplastic has been detected in food items such as commercial fish and chickens, fruits and vegetables, table salt and sugar, invertebrates like mussels, oyster, crabs, scallops, squid, and even in drinking water. And all of these food items is actually primary food source. And once we ingest these food items, we directly taken out microplastic into our body, and this microplastic will go into our GI tract. Another important thing that we need to know is that food packaging and food processing also may release microplastic into the food product. For example, during food processing, the producer use plastic-based machinery, plastic-based equipment and tools and this kind of machinery and equipment tools will release the MP during the processing until the final products. Another one thing is that food packaging. Plastic are released when the food containing plastic containers are heated or subjected to microwave. So this is how exposure through ingestion occur. Next, how exposure through inhalation can occur. The two most prevalent microplastic kinds found were fibers and sphere. But how fibers and sphere can be exist in our environment, in our air environment. So the source is synthetic microfibers like clothing, soft furnishing. Whenever the clothes are washed, fiber that comes from the cloth or any soft furnishing are discharged into the air and water. So this is how fibers and sphere can be accessed in our air environment. Another factor is various kind of face masks using during the COVID-19 also can be one of the factors how fibers and sphere can be exist in our environment. Another thing is human action like walking, crawling, cleaning the dust cause the already settled particle to get dislodged from surface and return into the air inside. For example, we walk or crawl on the carpet surface, floor surface, and during the cleaning or dusting the wall surface, it contributes to the release of fibers and sphere into our air environment. Next, how can exposure through dermal can occur? As we know that human skin has effective skin barrier, but then for the particles smaller than 100 nanometers still can cross this effective skin barrier. So when hands come in touch with contaminated surface or something that contains microplastic, this particle can stay and attach to the skin. So at the end of the day, it can cross the effective skin barrier. And human can has highest exposure when they use cosmetic that contains microplastic and personal care products like hand cleanser, facial and body scrubs, toothpaste. For example, we use shampoo, as we know that the scalp pore among the region where tiny particles are most likely to enter. And microbeads up to 40 nanometer could penetrate into epidermal Langerhans cells around hair follicle.
Assalamualaikum Doctor. My name is Siti Afifah Ramli. Now let me continue with the next part. In digestive system, microplastic ingestion may lead to inflammation or an altered intestinal microbiome, resulting in an imbalance between beneficial and harmful bacteria and potentially leading to symptoms like abdominal pain, bloating, and changes in bowel habits. Apart from that, the absorption of environmental toxins such as hydrocarbon and heavy metals carried by microplastics can lead to GI symptoms, including vomiting and abdominal pain. When inhaled, microplastics can induce oxidative stress in the airways and lungs, resulting in respiratory symptoms like coughing, sneezing, and shortness of breath, along with associated fatigue and dizziness due to decrease in blood oxygen levels. Furthermore, the primary mechanism of microplastic-induced neurotoxicity are acceleration of oxidative stress-induced damage and inhibition of the enzyme acetylcholine esterase. This enzyme plays an important role in maintaining normal nerve signal transmission. Figure 4.1 shows various harmful effects of microplastics on human health as discussed before. Now, let's move on to the metabolism of microplastic toxicity. Microplastic is degraded in the human body in the digestive system either by enzymes or bacteria. Common plastics such as polystyrene, polyethylene, polypropylene, PET and PVC have been found to be resistant to human digestive fluids. However, polystyrene microplastics can disrupt lipid digestion by interacting with lipids or lipases. When human colonic bacteria were present, PET microplastics experienced major surface alteration and structural degradation, affecting the colonic community composition. In addition, microplastics found in human feces may degrade in the digestive system channel. Figure 5.1 shows potential mechanism of metabolism and biodegradation of microplastics in human body. Number 1, microbiological activity in digestive system can degrade the microplastic. Number 2, microplastics may absorb organic compounds and be degraded due to exposure to physiologic fluids. Number 3, macrophage may release enzymes, acids and reactive oxygen species which degrade microplastic. And the last one is inflammatory reactions can lead to foreign body reaction, forming a fibrous encapsulation around microplastic particle. The incorporation of microplastic in the intestine occurs through three primary methods, which are adsorption, paracellular transport, and M-cell mediated transport. In adsorption, microplastics components cross the intestinal mucosa and are internalized by enterocytes through transcytosis. M cells initiates incorporation through various methods, including phagocytosis, pinocytosis, and receptor-mediated endocytosis. In paracellular transport, microplastics components are transported through gaps in villi, which aided by phagocytes. When microplastics enter the systemic circulations, they are transported to other organs, inducing adverse effects throughout the body. The intestine play a crucial role in overall human metabolism, including digestion, absorption, and immunity. Microplastics can disturb the natural balance of intestinal microbiota, thereby causing immune system abnormalities, intestinal inflammation, and abnormal metabolic processes. This disruption can promote the growth of dangerous bacteria, where compounds such as lipopolysaccharides can enter the bloodstream and harm other tissues and organs. Therefore, the impact of microplastics on the human intestine may have serious adverse effects on overall metabolism. Moving forward, we will cover mechanism of action of microplastic toxicity. In the human body, degradation of microplastics can occur both intra or extracellularly through two main mechanisms, hydrolytic or oxidative. Oxidative stress is one of the mechanisms of action of microplastic toxicity. Other oxidative stress-related antioxidants such as SOD, CAT, and glutathione peroxidase have been found to be disrupted in the liver, kidney, and gut of mice exposed to microplastics, resulting in an imbalance in energy and lipid metabolism. Second is inflammation. Inflammation is regarded to be a defensive mechanism that removes the source of cell damage and promotes tissue recovery, including due to harmful microplastic. Microplastics interact with fibroblasts and red blood cells, causing hemolysis and injury to the cell membranes and lead to systemic inflammation, contributing to COPD and asthma. Potential toxicity of microplastics in zebrafish found to reduce the number of Aeromonas and Pseudomonas and led to gut microbiota dysbiosis and inflammation. The interactions between microplastics and the immune system may have immunotoxicity and adverse effects including immunosuppression, immune activation, and abnormal inflammatory responses. The last one is neurotoxicity. Numerous studies have found neurotoxicity after exposure to microplastic both in vivo and in vitro. Microplastics in neurotoxic action by inducing necrosis process in erythrocytes, and the exposure to polystyrene microplastics in mice brains may affect the gene expression, promote neuroinflammation, and impair learning and memory. In human, microplastic toxicity can cause various signs of dysregulated nervous system as shown in figure 6.1. As a conclusion, the widespread and harmful impact of microplastics on various ecosystems require immediate attention. Defining the impacts of microplastics pollution are challenging due to the diverse conditions of the environment and multiple sources. Microplastics interaction with other contaminants, environmental conditions, and biological responses, making it difficult to recreate in a controlled laboratory context. To better understand the effects of microplastics, future studies should use standardized methodology, difference model, and interdisciplinary collaboration. Here are the list of references that have been used to prepare this presentation. Okay, that's all from us. Thank you for your attention.